Individual organisms died without fail of species continues to live through millions of years unless threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which species cannot survive for a long. Each individual leaves its progeny by a sexual or sexual mate. Sexual mode of reproduction enables creation of new variants so that survival advantage is enhanced. This unit explains the detail of reproductive process in flowering plant and human as easy to relate representative example. A related perspective on human reproductive health and how productive ill health can be avoided is also presented to complete our understanding of biology. Prachan Maheswari, born in born in 1904 in Jaipur, Rajasthan, Prachan Maheswari rose to become one of the most distinguished botanists not only for the India but for entire world. He moved to the Allahabad for higher education. He obtained DSc during his college days. He was inspired by Dr. W. Dodson, an American missionary teacher who developed in botany and especially morphology. His teacher once expressed if his student progressed ahead of him, it will give him a great satisfaction. This sports encourage Machan to what he teach in return. He worked on embryological aspects and popularized the use of embryological character in taxonomy. He established the Department of Botany, the University of Delhi as an important center of research of embryological and tissue culture. He also emphasized the needs of initiation of work on artificial culture of immature embryos. This culture has become a landmark of science in science. His work in test tube fertilization and intra-ovarian pollination won worldwide acclaim. He was honored with fellowship by Royal Society of London, Indian National Science Academy, and several other institutes. He encouraged general education and made a significant contribution to school education by his leadership, bringing out very first book of biology higher secondary book school published by NCERT in 1965. Sexual reproduction in flowering. Are we not lucky that plant reproduce sexually? The merits of a flower that we enjoy gazing at, the scents and the perfume that we swoon over, the rich color that attract us, all are this as an act to a sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. Looking at the diversity structure of the inflorescence flower and flower part shows an amazing range of adaptations to ensure formation at the end of a product sexual reproduction of fruits and seeds. In this chapter, let us understand the morphology and instruction and process of sexual reproduction in flowering plant. Angiosperm. Flower, a fascinating organ of angiosperm. Human beings have led an intimate relationship with flower and since time immemorial. Flower are object, aesthetic, ornamental, social, religious, and cultural value. They have always been used for uh, conveying important human feelings such as love, affection, happiness, gifts, mourning. Recently, five ornamental value of are commonly celebrated as homes and garden. Find out the names of the five more flowers used in a social and cultural celebration in your family. Have you uh, what does it refer to? To a biologist, flowers are morphological and embryological marble and sites of sexual reproduction. In early classes, you have various parts of a flower and will help you recall the parts of most important unit. Most important unit of sexual reproduction level. Ovary, sepal, filament, petal, anther, style, stigma. Pre-fertilization, extraction and event. Much before the actual flower is seen on the plant, the decision that the plant is going to flower has taken place. Floral hormone and structural changes are initiated. Floral hormone and structural changes are initiated which leads to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral and then the flower, the male and female reproductive structure, androecium, gynoecium differentiate and develops. You will recall that the endometrium consists of a wall of a stamen representing the male reproductive organ and gymnosium representing the female reproductive organ. 1.1 stamen, microsporidium, and pollen grain. Figure 1.2a shows the two parts of a typical stamen. The long and cylindrical structure called a filament, and the terminal generally bilobed structure of the anther. The proximal end of the filament is attached to thalamus or the petal of the flower. The proximal end of filament is attached to the thalamus or petal. थैलेमस या पेटल ऑफ द फ्लावर से अटैच होता है प्रॉक्सिमल एंड दैट इज फिलामेंट. फिलामेंट अटैच होता है थैलेमस या पेटल से. द नंबर एंड लेंथ ऑफ स्टेमेंस आर वेरिएबल इन फ्लावर ऑफ स्पीसीज. नंबर एंड लेंथ ऑफ स्टेमेंट डिफरेंट होता है, इन डिफरेंट स्पीसीज वेरिएबल होता है, अलग होता है. मतलब if you were to regrade the stamen each from 10 flower, each from species other species, and arrange them on a slide, you would be able to appreciate the large variation in size and nature. Careful observation of each stamen and good dissecting microscope and making neat diagrams would illustrate the range in the shape and attachment of the anther in flowering plant. A typical angiosperm anther is bilobed, with each lobe having two teeth, that is diathecus. Often a longitudinal groove runs lengthwise separating the. Let us understand the various types of tissue and their organization in the transverse section of anther. The bilobe nature of an anther very distinct in transverse section of the anther. The anther is a four-sided structure consisting of four microspheres located uh, at the corner, two in each lobe. The microsporidia develop further and become pollen sac. Microsporidia develop further and become pollen sac. They extend longitudinally all through the length of an anther and are packed with pollen grains. They extend longitudinally all through the length of an anther and are packed with pollen. Structure of microsporangia. In a transverse section, a typical microsporangium appears new circular in outline. It is generally surrounded by four walls layer. The the epidermis, endothelium, middle layer, and tapetum. The outer three wall layer perform function for protection and help in digestion of the anther to release pollen. The innermost wall of the is the tapetum. It nourishes the development of pollen grain. चार लेयर होते हैं, चार में से स्टार्टिंग के जो थ्री लेयर है एपिडर्मिस, एंथोलॉजी, मिडिल लेयर, वो परफॉर्म करते हैं फंक्शन फॉर प्रोटेक्शन एंड डिहाइड्रेशन ऑफ एंथर टू रिलीज पोलन. और जो टैप्टम है वो इनर मोस्ट है, वो नरिश करता है पोलन को. सेल ऑफ द टैप्टम पोजेस डेंस साइटोप्लाज्म एंड जनरली हैव मोर देन वन न्यूक्लियर. जो टैप्टम है उस डेंस साइटोप्लाज्म का होता है, जनरली मोर देन वन न्यूक्लियर होता है. When the anther is young, a group of compactly arranged homogeneous cells called sporangious tissue occupies the center of each microsporangium. When anther is young, a group compactly arranged homogeneous cells called sporangious tissue occupies the center of each microsporangium. Microsporogenesis. As anther develop, the cell of sporangious tissue undergo mitotic division to form microspore tetrad. What pleiad of the cell? Diagram filament, line of dehiscence, pollen grain, pollen sacs, anther. Tapetum, sporangious tissue, endothelium, epidermis, connective tissue, middle layer. Further, epidermis, endothelium, middle layer, microspore, mother cell, tapetum. pollen grain release हो रहा है. पहले in of microspore showing transfer wall layer, transfer section of a anther, a mature dehiscence and anther. As each of its cell of a spongious tissue is capable for giving rise to the microsporium tetrad, each one is the potential of pollen grain microscope mother cell. The process formation of microspores from pollen mother cell through meiosis is called microsporogenesis. As each of a cell of a sporangious tissue is capable of giving rise to the microscope tetrad, each one is a potential pollen or microscope mother cell. The process of formation of microspore from pollen mother cell through meiosis is called microsporogenesis. Microsporogenesis. The microspore, as they are formed, are arranged in a cluster of four cells. The microspore tetrad, as the anther mature and dehydrate, the microsomes dissociate from each other and develop into pollen grain. Inside each microsporangia, several thousand of microscope and pollen grain are formed that are released with the dehiscence of anther. माइक्रोस्पोर जो होते हैं वो टेट्रेट में एग्जिस्ट करते हैं, फिर वो फर्दर डेवलप होते हैं और वो पोलन ग्रेन बन जाते हैं. टेट्रेट से पोलन ग्रेन बनते हैं. Every spongy tissue have capacity to make formation of pollen mother cell. Pollen grain. The pollen grain represent the male gametophytes. If you touch the open anther of hibiscus or any other flower, you will find deposition of yellowish powdery pollen grain or your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will be really amazed at the variety of architecture, sizes, shapes, design seen on the pollen grain from different species. Pollen grain are generally spherical, measure about 25 to 50 micrometers in diameter. It has a prominent two-layer wall. The hard outer layer, called the exine, is made up of sporopollen, which is one of the most resistant organic matter known. The hard outer layer, called exine, is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperature and strong acid and alkali. No enzyme that degrades sporopollen is so far known. Pollen in exine has prominent apertures called germ pores where sporopollen is absent. Pollen grains are well preserved as fossils because of the presence of sporopollen. The exine exhibit a fascinating array of pattern and design. Why do you think the exine should be high? The inner wall of a pollen grain is called endine. It is a continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by the plasma membrane. Cytoplasm surrounded by plasma membrane. When the pollen grain mature, it contains two cells, the vegetative and generative cell. The vegetative cell is a bigger and has abundant food reserve and a large number of irregular shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperm, pollen grain are shared at this two cell state. In the remaining species, the generative cell divide mitotically to give rise to the two male gametes before pollen grain are shed. That is release stage. Pollen grain of many species is called severe allergy and bronchial affection. Pollen grain of many species cause severe allergies and bronchial affection in some people, often leading to the chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that come into India as contaminated with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Diagram: vacuoles, nucleus, asymmetrical vegetative generally. Pollen nutrient. It has become fashion in recent years to use pollen tablet as a food supplement. In Western countries, a large number of pollen products in the form of tablets and syrup available in the market. Pollen consumption has been taken claim to increase the performance of athlete and resource. When once they are shed, pollen grain have to land on the stigma before they lose the viability if they bring about fertilization. How long do you think the pollen grain retain viability? The period of which pollen grain may remain viable is highly variable. To some extent depends on the prevailing temperature and humidity. In some krill such as rice, wheat, pollen grain lose viability within 30 minutes to their release. And in some member Rosea, Leguminosae, Solanaceae, they maintain viability for months. You may have heard about the storing semen sperm animal, including human, for artificial insemination. It is possible to extract pollen grain of a large number of species of years in liquid nitrogen minus 196 degrees Celsius. Such stored pollen can be used as a pollen bank, similar to seed bank in crop breeding program. Pistil, macrospongia, ovule, and embryo. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpel, or may have more than one pistil, that is multicarpel. When there are more than one pistil may be fused together, syncarpel, or may create apocarpels. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform of a pollen grain. The style is elongated cylinder part beneath the stigma. The basal bulge part of pistil ovary. Inside the ovary is the ovarian cavity. Placental. The placenta is located inside the ovarian cavity. We recall the function and types of placenta that you have studied in class 11. Arising from the placenta are the microsporangia, commonly called ovule. The number of ovules in an ovary may be one. Example: wheat, paddy, mango, or too many: papaya, watermelon, orchids. Microsporangium ovule. Let us familiar ourselves with the structure and typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicles. The body of the ovule fuses with funicle in the region called hilum. हाइलम के रीजन में फनिकल से फ्यूज करता है ओव्यूल और फनिकल से प्लेसेंटा कनेक्ट होता है स्ट्रक्चर अटैच्ड टू द प्लेसेंटा बाय मींस ऑफ स्टॉक कॉल्ड फनिकल द बॉडी ऑफ द ओव्यूल फ्यूजेस विद funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Thus, hilum represents junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucleus except at the tip where a small opening called the micropial is organized. Opposite the micropial is a chalada representing the basal part of the ovule. Enclosed within the integument is a mass of a cell called nucellus. Cells of the nucellus have abundant trace of food material. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporangians. Enclosed within the integument is a mast cell called nucellus. Cells of nucellus have abundant food reserve material. Located in the nucellus of the embryo sac or female gametophyte, an ovule generally is a single embryo sac formed from a megaspore. Thalamus, ovary, style, stigma, carpels, hilum, funicle, micropylon, pole, outer integument, inner integument, nucellus, embryo, chalical pore. B is a multicarpellate, syncarpous pistil, papaver. Papavers, syncarpous, or multicarpellate. A multicarpellate, apocarpus, gynoecium, mycelia. C is mycelia. Apocarpus, syncarpus, apocarpus. Anatropous ovule. Megasporangiasis. The process of formation of megaspores from megaspore mother cell is called megasporangiasis. Ovules generally differentiate in a single megaspore mother cell in the microbial region. Microbial region of the nucellus. It is a cell containing dense cytoplasm and a prominent nucleus, the MMC. The MMC undergoes mutatic division. What the importance of MMC undergoing meiosis? Meiosis results in the production of 4 megaspores. Female gametophyte. In the majority of flowering plant, one of the megaspore is functional, while other 3 degenerate. Only the functional megaspore will develop into the gametophyte. Embryo. This method of embryo cell formation from a single megaspore is termed monosporic development. What will be the priority of the filament? 2 for 8 new question. Megaspore mother cell nucellus micropylon. Megaspore dad. Nucellus micropylon. मेगास्पोर टेट्राड माइक्रोपाइलट. फिर उसमें मतलब सिनर्जिड्स, बीच में वाला एक 2 सेंट्रल सेल, इसको बोलेंगे हम पोलर न्यूक्लियर. 3 एंटीपोडल सेल, ये फुल होगा synergids, synergid axis, central cell, polar nuclei, antibodies, challenge microbial form apparatus. Let us study about the formation of embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form two nuclei, which move to the opposite pole, forming two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of four nuclei and later eight nucleated of the embryo sac. It is interesting to note that these mitotic divisions are strictly free nuclei, that is, nuclear divisions are not followed immediately by cell wall formation. It is interesting to note that these mitotic divisions are strictly free nuclei, that is, nuclear divisions are not followed immediately by cell wall formation. After 8 nucleation, cell walls are let down, leading to the organization of a typical female gametocyte or embryo sac. Observe the distribution of a cell inside the embryo sac. 6 of 8 nuclei are surrounded by cell wall and organized into the cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. Called polar nuclei was situated below the egg apparatus. Egg apparatus के below the egg apparatus in the large central cell. There is a characteristic distribution of cells within the embryo sac. Three cells are grouped together at microbial end and constitutes egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have a special cellular technique at the microbiota called Filipa apparatus, which play an important role in guiding pollen tube into the synergid, which play an important role in guiding pollen tube in synergid. Three cells are at the chalazial three cells are at the chalazial end and are called antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at maturity, though 8 nucleate is 7 cell. 8 nucleate, 7 cell. Pollination. In preceding section, you have learned that the male and female gametes in flowering plants are produced in pollen gland and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain shared from anther to stigma is a pistil is termed as pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. Can you use a possible agent? Kind of pollination. Depending on the source of pollination can be divided into three types. Autogamy. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen from anther to stigma of the same flower. In a normal flower which opens and exposes the anther and stigma, complete autogamy is rather rare. Autogamy in such flower requires synchrony in pollen release and stigma receptive, and also the anther and stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis, and commelina produce two types of flower: chasmogamous flower, which are similar to flower of other species with apose, anther, and stigma, and clastogamous flower, which do not open at all. In such flower, the anther and stigma lie close to each other. When anther dehiscence in the flower buds, pollen grain come in contact with the stigma to effective pollination. Thus, clastogamous flowers are invariably autogamous, as there are no chances of cross-pollination landing on the stigma. Clastogamous flower produce assured seed set even in the absence of pollinator. Do you think classtogamy advantage or disadvantage of plant pie? Chasmogamous flower. Two types of flower are there. Chasmogamous. Viola, common pansy, oxalis, and commelina. Commelina. Viola, oxalis, commelina is an example of autogamy. 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 ये दो टाइप के होते हैं, जो कि क्लैस्ट्रोगैमस क्लोज, ये हेल्प करता है उसको, ठीक है। कैटोगैमस, transfer of pollen grain from anther to stigma of the another flower of the same plant. Although genetogamy, functionally cross-pollination involving pollinating agent, genetically it is similar autogamy since the pollen grain come from same plant. Xenogamy, transfer of pollen grain from anther to stigma of a different plant. This is only type of pollinator which during pollination bring genetically different. Only genetically different होता है xenogamy में. Agent of pollination. A is self-pollinated. B is cross-pollinated. C is castogamous flower. Agent of pollination. Plant use abiotic winds and water and one biotic animals agents to achieve pollination. Majority of plant use biotic agent for pollination. Only a small proportion of a plant use abiotic agent. Pollen coming in contact with the stigma is a chance factor in both winds and water for pollination. To compensate for this uncertainty associated loss of pollen, the flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst the abiotic pollination. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in the wind current. They often possess well exposed stamens so that the pollen are easily dispersed into the wind current, and the lark often feather the stigma to easily trap airborne pollen grain. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tessel you see are nothing but the stigma and style, which have waved in the wind to trap the pollen grain. Wind pollination is quite common in grasses. Uh, grasses and grasses and torn tessel cob is an example of wind pollination. Pollination by water is quite rare flowering plant and is limited to about 30 genera, mostly monocotyledon. And again, this you recall what water is a regular mode of transport for male gametes among the lower plant group such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because the need of water for the transport of male gamete and fertilization. ब्रायोफाइट्स और टेरिडोफाइट्स में की डिस्ट्रीब्यूशन बहुत लिमिटेड है क्योंकि उनको वाटर का फर्टिलाइजर के लिए ज़रूरत होती है, फर्टिलाइजेशन के लिए ज़रूरत होती है ताकि उसके मेल में ट्रांसपोर्ट हो सके. सम एग्जांपल ऑफ वाटर पॉलिनेटेड plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine seagrasses such as Zoastera, not such as water hyacinth and water lily. The flower emerge above the level of water and are pollinated by insects or bees, as in most of the land done. In Melisneria, the female flower reaches the surface of the water by long stalk and male flower pollen grain are released on the surface of water. They are carried passively by the water currents. Some of them eventually reach the female flower and stigma. In another group of pollinating plants such as seagrasses, female flower remain submerged in water and the pollen grain are released inside the water. Pollen grain in many such species are long ribbon-like and they carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grain are protected from wetting by mucilaginous covering. Mucilaginous covering से wetting करते हैं, ठीक है. Sea grasses में female flower submerged होती है. Pollen grain released inside the water. Pollen grain उस स्पेस में रिबन लाइक होता है. pollen grain ribbon like होता है, ठीक है. Pollen in many such species are long ribbon-like and they carry passively inside the water. Some of them reach stigma and achieve pollination. Both winds and water pollinated are not very colorful and do not produce nectar. Majority of a flowering plant range use a range of animals as a pollinator agent. Bees, butterfly, flies, beetles, wasps, and moths, birds, sunbirds, and hummingbirds and birds are the common pollinating agent. Among the animal, insects, particularly bees, are dominant biotic pollinating agent. Even larger animals such as some primates, lemurs, aboriginal tree dribbling, rodents, or even reptiles, gecko, lizards, and garden lizard, also been reported as a pollinator in some species. Often flower of animal pollinated plants are specifically adapted for a particular species animal. Majority of insect pollinated flowers are large, colorful fragment and rich in nectar. When the flower are small, when the flower are small, a number of flower cluster into the an inflorescence to make them constellation. Animals are attracted to the flower by color and are fragment. The flower pollinated by the flies and beetles secretes fall adults. The flower pollinated by flies and beetle secretes fall orders to attract this animal. To sustain animal visits, the flower have to provide rewards to the animal. Nectar and pollen are usual rewards, floral rewards. For harvesting the rewards from the flower, the animal visitors come in contact with the anther and stigma of the body. The body of the animal gets coating of a pollen grain, which are generally sticky in animal pollinated flower. When the animal carrying pollen on its body come in contact with the stigma, it brings about pollination. In some species, flower rewards are in providing safe places lay egg. An example is that the tallest flower, Amorphous, the flower itself is about 6 feet in height. In some species, flower rivers are providing a safe place to lay egg. An example is the tallest flower, Amorphous, 6 feet high. Okay, Amorphous. A similar relationship exists between the moth and plant of yucca, where both species, moth and plant, cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and flower in turn get pollinated by the moth. The larvae of the moth come out of the egg as a seed they start developing. Why Why don't you observe the sunflowers of the flowering plant or any other available you cucumber, mango, pepper, pepper, onion, lepia, cotton, tobacco, rose lemon, etc. Try to find out which animal visits them and whether they could pollinator. You will have to patiently observe the flower over a few days and at a different time. You could also recognize that see any relation between the characters of a flower and the animal you usually carefully observe. If any visitor come in contact with the other and stigma as only such visitor can bring pollination. Many insects may consume pollen or nectar bring pollination. Many insects may consume pollen or nectar without bringing about pollination. Such floral visitors are referred as pollen or nectar robber. You may or may not be able to identify pollinator, but you will surely enjoy their effort. Outbreeding device. Outbreeding device. Majority of a flowering plant produce hermaphrodite flower, and pollen grain are likely to come in contact with the stigma of the same flower. Majority of flowering plants produce hermaphrodite flowers, and pollen grain are likely to come in contact with the stigma of same flower. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and to encourage cross-pollination. In some species, in some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive or stigma becomes receptive much before the release of pollen grain. In some other species, the anther and the stigma are placed at different positions so that the pollen cannot come in contact with stigma at the same level. Both these devices prevent autogamy. Autogamy. The third device to prevent inbreeding is self-incorporation. This is a genetic mechanism and prevents the self pollen from same flower or other flower of the same plant from fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device prevents self-pollination is the production of unisexual flower. If both male and female flower present on the same plant, such as castor and match, that is monoecious, it prevents autogamy, but not geitonogamy. In several species such as papaya, male and female flower are present on a different plant, and each plant is either male or female dioecious. This condition prevents both autogamy and geitonogamy. Castor and match is monoecious. It prevents autogamy but not geitonogamy. Pollen pistil extraction. Pollination does not guarantee the transfer of a right pollen, compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the species form or from same plant, if it is itself incomplete, also land on the stigma. The pistil has the ability to recognize the pollen whether it is a right type or wrong type. If it is the right type, the pistil accepts pollen and promotes the post-pollination events that lead to fertilization. If a pollen is a wrong type, the pistil rejects pollen by preventing the pollination or germination on the stigma. The pollen growth in the style. The ability of the pistil to recognize the pollen followed by the acceptance or rejection, the result of a continuous dialogue between the pollen grain and pistil. This dialogue is mediated by chemical component of the pollen interacting with those pistil. It is only recent years that the botanists have been able to identify some pollen and pistil component and interaction leading to the recognition followed by the acceptance or rejection. As mentioned earlier, the following compactal pollination, the pollen grain they meet on the stigma to produce pollen tube through one of the jumper. The pollen grain germinates on the stigma. Pollen grain germinate on the stigma to produce pollen tube through one of the jumper. The content of the pollen grain move into the pollen tube. Pollen tube. Diagram. A diagram is the pollen grain germinating on the stigma. B diagram pollen tube growing through style. L is longitudinal section of a sowing path of pollen tube growth. Longitudinal showing pollen grain antipodal polar nuclei excel synergies. Longitudinal section of flower showing pollen. T is एक synergy change in territory वही मेल गैमेट और वेजिटेटिव सेल है फॉर्म apparatus. मेल गैमेट आगे होता है वेजिटेटिव पीछे होता है synergids. प्लाज्मा एक न्यूक्लियर सेंटर मेल गैमेट का pollination of polar nuclei. Discharge of a gametes into the synergy. Sperm one into the egg and like the other into the center. Pollen tube goes through the tissues of the stigma and style and reaches the ovary. You will recall in some plant pollen grain are shed at two cell condition, a vegetative and a generative cell. In such plant, generative cell divide into form two male gametes during the growth of a pollen tube on the stigma. In plant which shed pollen in the three cell condition, pollen tube carry two male gametes from the beginning. Pollen tube after reaching the ovary enters the ovule through the micropyle and enters one of the synergy through filiform apparatus. Many research shows that the filiform apparatus is present in the micropylar part of the synergids guides the entry of a pollen grain. Synergids guides the entry of pollen grain. All these events from pollen deposition to the stigma until pollen enter the ovule are together referred as pollen extraction. pollen के डिपोजिशन से ओव्यूल के जाने तक हम उसको कहेंगे pollen pistil extraction. As pointed out earlier, Together, pollen pasteurization is a dynamic process involving pollen recognition followed by promotion or inhibition of pollen grain. Pollen pasteurization is a dynamic process because process involving pollen recognition followed by promotion or inhibition of the pollen grain. The knowledge gained in this area would help the plant breeder in manipulating pollen pistil interaction, even in compactile pollination to get desired hybrid. You can easily study pollen germination by dusting of some pollen flowers such as pea, chickpea, Carteria, balsam, vinca on a glass slide containing a dropsable solution. After 15 to 30 minutes, observe the slight low power lens of the microscope. You will likely to see pollen tips coming out of the pollen grain. A breeder is interested in crossing different species and often genera to come and desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach of one of the major approaches of crop improvement program. Artificial hybridization is one of the most improvement program. In such crossing experiment, it is important to make that only desired pollen are used for pollination and stigma is protected from contamination from unwanted. This achieved by emasculation or bagging techniques. In the female parents pair bisexual bird, removal of anther from the flower bird before the anther dehisces using a pair of forceps as necessary. This step referred to emasculation. Emasculation is the removal of removal of male anther. Emasculated flower have to be covered with a bag of a suitable size, generally made up of butter paper, to prevent contamination of stigma with unwanted pollen. This process is called bagging. When the stigma of a bag flower attain a receptively moisture point and then collected from the anther of the male parent are dusted on the stigma and the flower are rebagged and the fruits allowed to develop. If the female parent produce unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and flower rebag. If the if the female parent produces unisexual flower, there is no need for emasculation. The female flower buds are bagged uh, before the flowers open. Double fertilization. After entering the one of the synergy, the pollen tube releases uh, two male gametes. After entering out of the synergid. After entering one of the synergids, the pollen tube releases two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the axil. And fuses with its nucleus, thus complementing the syngamy. This results in the formation of diploid cell zygote, and the other male gametes move towards the two polar nuclei located at the central fuse and fuses with them to produce triploid primary endosperm nucleus, triploid PEN, primary endosperm nucleus. As this involves the fusion of three haploid nuclei, it terms triple fusion. Since two types of fusion, syngamy and triple fusion takes place in embryo cell. The phenomenon is termed as double fertilization and even unique in plants. The central cell triple fusion becomes primary endosperm (PEC). The central cell after triple becomes primary endosperm cell (PEC) and develops into the endosperm, while zygotes develop into an embryo. while zygote embryo बनेगा, primary endosperm बनेगा. Degenerating cell. Degenerating synergids, zygote to an primary endosperm cell, primary endosperm nucleus, degenerating antipodal cell. से जाइगोट बनेगा, जाइगोट से ग्लो बनेगा, फिर ग्लोबियर एम्ब्रियो बनेगा, फिर ग्लोबियर हार्ट शेप एम्ब्रियो बनेगा, और ऊपर वाला सस्पेंसर रहेगा, ऊपर वाला स्ट्रिया, नीचे प्लूमेल बनेगा, रेडिकल कॉटिल्यूडन मैच्योर. Fertile embryo shape showing zygote and primary endosperm nucleus. Pen. Stages and of the embryo in a zygote shows in a reduce as compared to the A. 1.4 Post-fertilization structure and habits. Flowering flow following double fertilization, the event of endosperm and embryo development, maturation of ovule into the seeds and ovary into a fruit collective term as post-fertilization. Then endosperm. Endosperm development precedes embryo development via uh, the primary endosperm cell divides repeatedly and forms a triplet. Endosperm tissue. The cells of these tissues are filled with reserved food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the PEN, primary endosperm nucleus, undergoes successive nucleus division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from a tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei and surrounding while kernel is cellular endosperm. फ्री न्यूक्लियर डिफिजन का एग्जांपल है वाइट कोकोनट, टेंडर कोकोनट और वाइट कर्नल जो होता है वो सेलुलर एंडोस्पर्म का एग्जांपल है. एंडोस्पर्म में either be completely consumed by the developing embryo, pea, groundnut, beans before seed germination, or it may be persist in the mature seeds. Example castor and coconut. कैस्टर और कोकोनट में मैच्योर सीड में भी रहता है एंडोस्पर्म. तो ये होगा नॉन एंडोस्पर्मिक, ये होगा एंडोस्पर्मिक. एंडोस्पर्मिक इज कैस्टर एंड कोकोनट एंड बी यूज्ड एज ड्यूरिंग seed germination. Split upon some speeds of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in krill, wheat, and rice, maize. Embryo. Embryo develop at the microbial end of the embryo sac where the zygote is situated. The most zygote divides only after a certain amount of endosperm is formed. Most zygote divides only after a certain amount of endosperm is formed. Zygote is situated embryo developed at the microbial end of the embryo sac where the zygote is situated. Most zygote divide after uh, uh, certain amount of endosperm is formed. This is an adaptation to provide assured nutrition of the developing embryo. Though the seed differs greatly, the early stage of embryo development embryo journey are similar in both. Monocotyledon and cotyledon depicts the stages of embryo journey in a dicotyledon embryo. Zygote gives rise to the uh, pro-embryo and subsequently globular, heart-shaped and mature. Pro-embryo, फिर globular, फिर heart-shaped, फिर mature. गोलू, फिर गोलू का इंतजार किया. A typical dicotyledon consists of an embryonal axis and two cotyledons. Embryonal axis or two cotyledons. The portion of embryonal axis above the level of a cotyledon is the epicotyle. एक्सिस के अबो लेवल पे जो होता है कोटिल्यून के वो एपिकोटाइल और विच टर्मिनेट्स इज अ प्लूम्यूल. एपिकोटाइल ऊपर वाला, नीचे वाला प्लूम्यूल. फर्स्ट टाइम the the slender portion below the level of a cotyledon is hypocotyl. Portion below the level of cotyledon के नीचे hypocotyl that terminates to it at its lower and end in the radical or root tip. The root tip is covered by root cap. Embryos of monocotyledon possess only one cotyledon in the grass family. In the grass family, the cotyledon is called scutellum. Grass family, the cotyledon embryos possess only one cotyledon in grass family. Cotyledon is called scutellum. That is situated towards the one-sided lateral or the embryonal axis. At its lower end, the embryonal axis has radical root cap enclosed in an undifferentiated sheet called chlorhiza. The portion of the embryonal axis above the level of attachment of the scutellum is the epicotyle. Apicotal has a shoot apex and few leaf pyramidal enclosed in a hollow follicular structure called coleptile. Soak a few seeds in a water, say wheat, maize, peas, sesame, overnight, then spread the seeds and observe the various parts of the embryo. ऊपर प्रोम्यूल, बाजू में कॉटिल्यूड, ये दो कॉटिल्यूड, नीचे वाला पार्ट हाइपोकॉटाइल, ऊपर वाला प्लूम्यूल, उसके नीचे रेडिकल, रेडिकल के नीचे रूट कैप और मोनोकॉटिल्यूड में स्क्यूटेलम, जो कॉटिल्यूड बन रहा है उसको बोलते हैं कॉलेप्टाइल, जहाँ से जर्मिनेट हुआ, शूट एपेक्स, एपेक्स में एपीब्लास्ट, रेडिकल, रूट कैप और सबसे नीचे वाला पार्ट क्लेरोराइजा, मियोग्रास, डाइकोटिंग्रो। कॉलेप्टाइल का है, एपिकोटाइल, look at coleptile ऊपर, coleptile होता है मोनोकॉट। In angiosperm, the seeds is final product of sexual reproduction. It is often described as the fertilized ovule. Seeds are formed inside the fruits. Seeds are formed inside a fruit. A seed is typically consists of a seed called cotyledon and embryo axis. The cotyledon of the embryo are simple structures, generally thick and swollen due to the storage of food reserve legumes. Mature seeds may be non-albuminous or X-albuminous. Non-albuminous seeds have no residual endosperm as it's completely consumed during embryo development. Example pea, groundnut. Albuminous seeds retain a part of endosperm and is not completely used up during embryo development. Wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnants of nucleus are also percented. This residual percentage nucleus is the perisperm. Integument of ovule harden and stop protective seed coat. The micropyle remain as small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the integument of ovule harden and stop protective seed coat, the micropyle remain as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As seed matures, its water content is reduced and seed becomes relatively dry, 10 to 15% moist by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactive called dormancy, or if favorable conditions are available, advocate moist oxygen and suitable temperature, they germinate. As ovule mature into seed, the ovary develop into the fruit. That is the transformation of ovule into the seed, and ovary into the fruit proceeds simultaneously. Ovule mature into the seed, the ovary develop into the fruit. That is, the transformation of ovule into the seeds and ovary into the fruits proceeds simultaneously. The wall of the ovary develop into the wall of a fruit, called pericarp. The fruits may be fleshy in guava, orange, mango, etc., or may be dry, as in groundnut and mustard. The fruits may be fleshy, guava, mango, orange, and may be dry, groundnut, and mustard. Many fruits have evolved mechanism for dispersal of seed. Recall the classification of a fruit and their dispersal mechanism that you have studied in earlier class. Is there any relationship between the number of ovules in an ovary and the number of seeds present in the fruit? In most plants, by the time of the fruit developed from the ovary, the other floral plant regenerate and the follow. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contribute to fruit formation. Such fruits are called false fruit. Most fruit, however, develop only from ovary and are called show fruits. Although in most of the species, fruits are result of the fertilization, there are few species in which fruit develop without fertilization. Such fruits are called parthenocarpy fruit. Example banana. Banana is one such example. Parthenocarpy can be included in the application of a growth of hormone, and such fruits are seedless. Cotyledon. Figure A structure of some seeds. Cotyledon, micropyle. Seed coat, endosperm, cotyledon. Hypocotyle axis root tips. ऊपर वाला पेरिका, अंडर वाला एंडोस्पर्म, स्क्यूटेलम, कॉलियोप्टाइल, क्लोराइजा, रेडिकल, 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 स्क्यूटेलम, कॉटिलेडॉन, सीड कॉर्ड, शूडापिकल, हाइपोथैलेमस, एंडोस्पर्म, मेसोकार्प, एंडोकार्प, सीड थैलेमस, थैलेमस, Seed offers several advantages to endosperm and angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have a better adaptive strategy for disposal of new habitat and help in the have to colonize the other areas, as they have sufficient food reserve and young seedling and nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection of the young embryo. Being product of sexual reproduction, they generate new genetic combination leading to the variant. Seed is the basis of our agriculture. Dehydration and dormancy of mature seeds are crucial for storage seeds, which can be used as a food throughout the year and also rise the crop in the next season. Can you imagine the agriculture in the absence or presence of a seeds which germinate straight away soon after formation cannot be stored? How long do the seeds remain alive after they disperse? This period varies greatly in a few species. Within a few months, seeds of large number of species live for several years. Some seeds can remain alive hundreds of years. There are several records of very variable origin. The oldest is lupin, Lupinus architectus, evacuated from Arctic tundra. ओल्डेस्ट सबसे ओल्डेस्ट जो हमारे पास है वो है ल्यूपिन आर्कटिक टुंड्रा से सीड जर्मिनेटेड एंड फ्लावर्ड आफ्टर एन एस्टिमेटेड रिकॉर्ड 10,000 ईयर ऑफ डोरमेंसी 10,000 आर्गेनिक्स का a recent record of a 2,000-year-old viable dead palm phoenix declifera डिस्कवर आर्कियोलॉजी में किंग खोयर पैलेस 10,000 साल का ल्यूपिन किंग टोंड्रा में और डेड पाम फोइनिक्स डेक्टिलाइफ्रा डिस्कवर्ड 2000 पहले का इन किंग प्लैनेट के पास आफ्टर कंप्लीटिंग अ ब्रीफ अकाउंट ऑफ सेक्सुअल फ्लावरिंग प्लांट इज वर्थ अडॉप्टिंग अ कंप्लीट by asking how many eggs how many orchids fruits are one such category. Each fruit contains thousands of tiny cells. Can you think some plants in which fruits contain very large number of seeds? Orchid fruits are one category. Orchids में large number of seeds रहते हैं. Each fruit contains thousands of tiny seeds. Similar is in case of a fruit of some parasitic such as Aubrinia or Astrina. Have you seen a tiny seeds of a ficus? How large is a tree of ficus formed tiny seeds? How many billions of seeds does ficus reproduce? Can you imagine other example in which tiny structure can produce a biomass? Other seeds in general seed in the general product of fertilization. A few flowering plants such as some species Astraceae and grasses. Astraceae and grasses have evolved a special mechanism to produce seed without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several waves of development. Apomictic cells in some species, diploid sex cell is formed without reduction division and developed into the embryo without fertilization. More often is as many citrus and mango varieties of some Astraceae और classes के सब प्लांट में स्पेशल मैकेनिज्म है to produce seed without fertilization के बिना हो जाता है वो होता है apomixis Astraceae और classes में apomixis होता है in some species diploid egg cell is formed without reduction division and development in the embryo without fertilization. More often as many in citrus and mango varieties of the nucleus cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into the embryo. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shape. Count the number of embryos. What will be genetic apomixis embryos? Can they call tons? Hybrid varieties of several of our food and vegetable crop are being extensively cultivated. Cultivation of hybrid has determined increased productivity. One of the problem of hybrids that is hybrid seeds have to be produced every year. If the seed collected from the hybrid are sown The plant in the progeny will segregate do not maintain hybrid character. The production of hybrid seeds is costly, hence and the cost of a hybrid seed become too expensive for the farmer. If these hybrids are made into apomix, there is no segregation of a character in the hybrid progeny. Then the farmers can keep using hybrid seeds to raise new crop year after year, and he does not have to buy hybrid seeds every year because of important of apomixis in hybrid seed industry. Active research is going on many laboratories around the world to understand genetic apomixin and transfer to the apomictic genes into the hybrid varieties. Summary: flower seed of flower site of sexual reproduction in organism. In the flower, androgen of stamen represent the male reproductive organ, and androgen condition of pistil represent the female reproductive organ. Atypical, and there is a biolog diatheticus tetraspermia pollen developed inside the microsporidia foliar wall. There and septum surrounded the microsporidium cells of the exponential line in the center of the microsporidium undergo meiosis to form tetrads of microspore or individual microspore mature into the pollen, and pollen represents the male gamete of a generation, and the pollen in two layer, and the outer exine and the exine is made of has a jumper pollen may have a vegetative and generative three some vegetative and two male gamete at the time of shedding. Pistil has the pistil has three parts: the stigma, style, and ovary. Pollen grain may have two cells or three cells. As remember, the pistil has three parts: stigma, style, and obviously represent the ovary. The ovary is structural productive, and an opening is microscopic. The central tissue of the nucleus is which are can differentiate a cell. A cell of the megaspore mother cells divide mitotically, and one of the megaspores form embryo cell. The female modified mature embryo 7 8 cells in the micropyle, and the egg apparatus consists of two synergies and an egg cell. The at the challenge and three anticotyl cells. The center is the large at central with two polynuclear pollination. Pollination is a mechanism of transfer of pollen from ancestors by pollination. Pollination is either abiotic or abiotic. Pollen pistol instruction involve all events of the landing of pollen to stigma until the pollen tube enters in the embryo sac, the colony or pollen inhibition. And pollen compatible pollen grain germinate on the stigma and settling the pollen tube or ovarian ovules and finally discharge two male gametes and one woman. Angiosperm exhibit double fertilization because of two fusion events occur in embryo sac, namely syngamy and triplet. The product of this fusion are zygote and triploid primary endosperm nucleus. That is in primary endosperm zygote. Zygote develops into the embryo, and primary endosperm cells form endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo or globular heart-shaped stage before maturation. Mature dichotomy. The embryo has two dichotomy and one embryonal axis with apicotyl hypocotyl. Embryos of monocotyledon have a single cotyledon. After fertilization, ovary develop into the fruit, and ovary develop into a phenomenon called apomixis found in some angiosperm, particularly in grasses. It results in the formation of seed without fertilization. Apomixis have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seeds. This phenomenon is called polyembryony.